So the hippocampal formation is a brain region that is involved in learning and memory. And our fundamental question is how exactly the brain processes information, processes and stores information. Okay, so that's our fundamental question that we ask. Okay, let everybody settle down so that you can focus on the information here rather than the information there. Alright, so we use a combination of techniques. We use spatial electrophysiology where we record from individual neurons. So that's how hippocampal neurons look like under the microscope. We also do in vivo field recordings from rodents, recording from different parts of the hippocampus. And we do a lot of computational neuroscience, both at the single neuron level and the network level, basically. So that's broadly the kind of techniques that we use. And predominantly we work at the cellular level of things, but we also dabble into the molecular and network end of it, a little bit into the behavioral end of it, especially for the in vivo part. So for today's talk, I thought I'll take you through the 4 generations of neuronal models spanning over more than a century actually. So it's not common knowledge, at least I come from an engineering background, did my PhD in engineering and then moved over to neuroscience. So I know that it's not common knowledge in the engineering side of it to recognize that there are generations of single neuron models and it has matured way beyond what it was in the 1900s basically. So several deep learning models still continue to use the perceptron as the fundamental model of how exactly neurons work. So I thought I'd take you through the different generations of single neuron models and what exactly gave rise to these different models, what was the changing invention / discovery that led to that generation. But I'll just take you through these. So this is first generation. So it all started with Cahal, as always. He's considered as the father of modern neuroscience. So that point of time there was a raging debate between Ramoni Cahal and Golgi about the difference between what we called as neuron doctrine versus the reticular theory. So the neuron doctrine is the one that proposed that the nervous system is made up of discrete individual cells, which would be neurons, which was Cahal's framework, and the reticular theory idea. Reticulum basically stands for a network, and there the nervous system was considered as a continuous meshwork of cytoplasm. There is a little bit of truth to both of them, even though we think that the neuron doctrine as one, there are cells which are connected by gap junctions, and therefore there's a little bit of truth here also. So one of the funniest things is that for showing that there are individual cells, Cahal actually used Golgi's staining technique, and because of the fact that it was sparse labeling technique, he was able to identify single neurons. If you search for Ramoni Cahal on Google, you will see that there are several tracings of him, beautiful drawings from him of single neurons showing that these are separate neurons. So it's where it started. So even if in their Nobel lecture, you will see that they would fight with each other at that point of time. So it was Cahal who proposed the idea of the so-called law of dynamic polarization, which simply states that information flow in a neuron is unidirectional. The dendrites are the ones that receive the information, and there is flow of information from the dendrites to the cell body where it is kind of integrated, and there is a single spike initiation location which is called as the axonal initial segment, where spikes get initiated depending upon what exactly the integration that occurs over here, and eventually the spike traverses through the axon to reach the other dendrites through synapses that are present over here. So that's one of Cahal's drawings. Based upon this knowledge that there are different neurons, there are dendritic structures, there's an axonal initial segment. It was Lapik, I'm 100% sure I'm not pronouncing it right, but I think it's okay. So he proposed a long time ago in 1907 the integrated file model. So this was way back in 1907. It was at that point of time that the integrated file model was proposed. It's a picture from his paper where there is a parallel RC circuit that is present over here. And the idea here is that this RC circuit will integrate information with a certain time constant. In neurons, that number is around 30-40 milliseconds, the hippocampal cortical neurons. And then there is a threshold for the voltage. Once the neuron reaches, or this model reaches that particular threshold, you would elicit an action potential. And therefore, it's an integrate and fire model for how exactly a neuron functions. So in today's world, you would look at it as that's a membrane. There are different kinds of protein molecules which are called as ion channels, which would act as models for the resistance. More the number of ion channels present, the lesser the resistance or the higher the conductance. And there is a capacitance because there is a ionic conducting media on this side, ionic conducting media on this side, and there is a dielectric sitting in between, and that acts as a capacitance. So you would have, if you solve, if you use simple Kirchhoff's current law, you would arrive at something like this, giving you the differential equation for voltage as a function of the components here, as well as the synaptic current that comes through. So in this model, what you would do is that if you are injecting a certain constant current injection, let's say, you would see that the neuron charges up to a certain threshold voltage, and then you would artificially reset it, and the reset would bring it back to resting membrane potential after a certain refractory period, and the process would continue over and over again. In this case, it is current injections, which are pulse current injection. You could have synaptic inputs, you could have excitatory, inhibitory synaptic inputs, and that would give you the integrated fire model for how exactly neurons function. A more discrete time version of this is the perceptron. Here the integration is replaced by a simple summation, and there we had a threshold element. Here there is a nonlinearity that sits over here, which says that if this summation crosses a certain threshold, you would elicit an action potential basically. So this is fundamental discrete version of the big model. And here you would have F as the nonlinearity, and it implements some kind of thresholding, and you have several different ways of implementing it. Fundamentally, these are integration fire structures, one way or the other. So it started out in 1907, and this, as I said, is the predominant model that people think how neurons function, but this is absolutely wrong, to say the least. So there are different generations, different variations to these Gen 1 models in terms of the different activation functions of the perceptron-like model, or you can have different kinds of integrating fire model where you can have bursting behavior, you can have spike frequency adaptation. By having these couple differential equations and having some kind of an artificial thresholding element and a reset mechanism, you would have different kinds of spiking neurons in the structure. Right? So basically this is the first generation, 1900s. The technical innovations that guided this were the Golgi staining technique, without which Kahar wouldn't have known that there are individual neurons, and membrane biophysics, which led to the RC circuit formulation, and the conceptual foundations were the neuron doctrine, the law of dynamic polarization, because this is essential because it lets you assume that there is unidirectional flow of information, and fundamentally the concepts from electric circuits moved in here, and in the continuous time version it would be an ordinary differential equation. In the discrete time version it would be an algebraic summation. It's a generation. The second generation started in 1950s. So the question that was asked in this generation was the question of where exactly is that nonlinearity coming from. In all the integrating fire models, the threshold was artificially imposed, whereby you say that if it reaches beyond this threshold point, you elicit an action potential. But the question is, where is the origin of this? What exactly is giving you that threshold? Is a fundamental question. And by the time, Hodgkin and Huxley and Kenneth Cole and others started recording from the squid giant taxon, and they knew that action potentials look like this. Right? There is a threshold voltage, That is, you are injecting a certain amount of current, and that current is integrated by the RC, let's say, and once you reach a certain threshold voltage, you elicit this action potential, which is a 100 millivolt signal with 1 millisecond width, and that is the action potential basically. So the fundamental question that Hodgkin and Huxley asked was, what exactly is the origin for this threshold voltage? How do you get this 1 millisecond signal as a consequence of you crossing this threshold voltage? Right? So one of the fundamental technical innovations that led to Hodgkin and Huxley, which did not get as much recognized as Hodgkin and Huxley got recognized, is Kenneth Cole's work. He was the one who invented the voltage clamp amplifier. So the idea is that in an electric circuit you can either inject a current and measure the voltage, or you can maintain the voltage to be constant and measure the current basically. But for maintaining the voltage to be constant, you need a feedback circuit, right, which is really fast, and you are basically continuously comparing it with a command voltage to which you want to clamp it to, measure the voltage from this quick giant taxon, and keep comparing it and ask questions about what that difference is, inject a current back into it, which will nullify whatever transmembrane current is coming through this. So this was an important technical innovation without which Hodgkin and Huxley wouldn't have been able to do the kind of experiments that they did. So and go through the 5 papers of Hodgkin and Huxley. That is field changing. Even today, that is pretty much the poster child for computational neuroscience, for electrophysiology, for physiology as a whole. So please go through those 5 papers that were published in 1952 in J Physiology. And it was Hodgkin and Huxley who figured out using the squid giant taxon. Why is it important that they chose the squid giant taxon? Because the squid giant taxon is almost a millimeter in diameter compared to our axons, which is on the order of 2 microns. So they had to send this cable through the axon for them to be able to record this, and therefore choosing the right system was an important part of figuring out how exactly that action potential got generated. So in the 1950s, they got their Nobel for this with John Nichols. And it turned out that the action potential was mediated by ionic movement, unlike the electrical system where it is electronic movement that causes voltage changes and stuff like that. Here it is ionic movement that causes changes in membrane potential differences between the inside of the cell and the outside of the cell. So what is shown over here is some kind of a machine that sits over here, which is dependent upon voltage difference between the inside and the outside. And if the voltage crosses a certain threshold, then this machine starts letting in a specific kind of ion. Say for instance, this would be a sodium channel, and it allows only sodium to pass through, and it allows it to pass through only when the voltage crosses a certain threshold. And that is where the nonlinearity comes from basically. The voltage dependent properties of this nano machine, if you will, is what is giving you the voltage dependence that you observe for a neuron to elicit an action potential. So, in a nutshell, what Hodgkin and Huxley showed was there were channels. The sodium channel has a voltage dependence like this. That's the membrane voltage, the difference between the inside and the outside, and the probability of sodium channel opening. And you see that as you increase the voltage from -80 to +60, you see that there is an increase in the open probability. And the threshold for opening is around -50 or -40, which is also the threshold for action potential generation. So what happens is, once you reach this point, you will see that the sodium channels start opening, and there is a fast positive feedback loop that drives the influx of sodium into it. Where does the positive feedback loop come in? You open a little bit of sodium channels, you get more sodium into the cell. That's a positively charged ion. That further depolarizes the cell and therefore takes it here. The probability goes up further. And therefore, there is a huge influx of sodium into the cell that takes it towards what is known as the equilibrium potential for sodium. And eventually what happens The sodium channels have a mechanism to shut themselves down by a process that is called as inactivation. So the sodium influx comes down and this depolarization also opens up potassium channels, and unlike sodium, when potassium channels open, the potassium ion goes from inside to the outside, and therefore you will have positively charged ions going out, and that brings the voltage down and it goes back to where it was. And the millisecond duration is a function of the difference in the kinetics of the sodium and the potassium channel that sits over here. And this gave us an idea of, it took away the mystery about where exactly the threshold was coming from. It told us clearly that there was a specific set of channels that were present which gave us this kind of an action potential basically. Right? So and this, the beauty of Hodgkin and Huxley is not just that the kind of experiments that they did, the kind of system that they chose were par excellence, but also they took that data, sat on for 4 long years, put together a model, fit the parameters. That's the system of differential equations that define what exactly the Hodgkin, the action potential is. And so we started out with this RC circuit from the integrating fire era. Now what we have done is we have added 2 more parallel conductances to this with specific kind of voltage dependent properties. Right? So this N infinity of V is the voltage dependence of the potassium, these 2 correspond to the sodium, and this is just simple KCL on that node, and you will get this kind of an equation. And this is the system of differential equations that defined the onset of the second generation of models for how exactly the neuron functions. Additional parallel conductances to this and find out how exactly you get different kinds of neurons firing with different kinds of patterns. These are all variants of this, and the Hodgkin and Huxley model also gave the foundation for not just neurons but also for other excitable cells like hearts, muscles, the kind of channels that are present over there, the kind of action potentials that are elicited there. All of that started from the Hodgkin and Huxley model, which, as I said, is in the second generation of neuronal models basically. So that's 1950s. The voltage clamp circuit is an important technical innovation. Without this, they wouldn't have been able to measure the ionic currents. They recorded from the squid giant axon. That's an important detail because they are an wide, and the ionic current interactions and the voltage dependent nonlinearity made the conceptual foundations here. And instead of having one single differential equation there, here we have a coupled system of differential equations now. Some of them defining the ionic currents, some of them defining the voltage, and they are coupled to each other. And it brought in the nonlinear dynamics era of neuronal models into neurophysiology. The third generation of models brought space integration. Right? I mean, so far it was considered that a neuron is some kind of a sphere, and you have all these different ion channels and the membrane components present over here in that particular sphere. But in a real neuron, the voltage here and the voltage here are not exactly the same, and therefore you can't even write this kind of an equation because this is a simple ordinary differential equation where V is a function of only time. In this scenario, V is not just a function of time, V is also a function of space as well as time, and therefore you will have to start with partial differential equations. So, and the second thing will come to this later. So how do we even start with the understanding the idea that okay, there are different compartments in a neuron, each one can have different voltages. How do we put together a model for this? It turned out, Wilfred Roll, who will come here, figured out that you could use the cable equation, which was derived by Lord Kelvin a long time ago for laying the first transatlantic telegraphic cables. So this is all like 1800, 1900s. So it was used to compute the attenuation of signals in a telegraphic cable. So what Wilfred Roll figured out was that you could use the same equation for assessing how exactly a voltage signal that starts at one point, how exactly it reduces, how exactly it attenuates as a function of distance, and can we have models for the components that are present in terms of the ion channels, in terms of the intracellular resistance offered by the cable, and so on and so forth. So that's the cable equation. For those of you who used the diffusion equation, the Vm term wouldn't be there. So it's the cable equation. It's a second order partial on distance, first order partial on time, and there's tau and time constant, which I said was 30-40 millisecond, which is R into C, and the lambda is what is called as the space constant. It defines how exactly the signal attenuates across distance. So in this model, the diameter is accounted for. The intracellular resistance offered by the ions moving inside. It is electrodiffusion, and you also have different organelles that are present. And there are these transmembrane currents, the resistances and capacitances, and even the ionic currents could be accounted for in this particular scenario. Right? So this is where the third generation of models took inspiration from. But the fundamental technical innovation that changed the whole thing in the early 90s, where these two people who came up with this technique called as the patch clamp electrophysiology. It was first designed as a mechanism for recording single molecules, the function of single molecules, the ion channels that we talked about. So they are binary in nature for most part. And you will see that they either open or close. Here there are two different ion channels that are present. So this current is on the order of 2 or 3 picoamperes basically. I mean that's the kind of number that we are talking about. So these, it's a glass pipette, there is a specific solution inside, and you can record the function of this single ion channel using this particular technique. So the paper came in 1982, the ion channel patch clamp physiology, but this one, which allowed us to visually look at specific locations of a neuron and patch them at either the cell body or at the dendrites. So here there are two different electrodes. This came from Bert-Sackmann's lab, and this one was in 1993 basically. So this was the first time that we were actually able to record from the dendrites. Until that point of time, we just assumed that the dendrites' fundamental function was just to collect the information from here and send it down this particular structure where the action potential is initiated. But this was the first time that we were able to actually record from neurons here the actual voltage signal, and we could also do cell-attached recordings like this and find out what kind of ion channels, if any, are present over here basically. Right? So that was the technical innovation that led to the third generation of model where space was brought into the picture. If the dendrites were simply passive, they did not do anything else, the earlier models would have completely held up. But it turned out that as people started doing these recordings, so this is actually a hippocampal pyramidal neuron now, and people recorded from the dendrites, performed cell-attached recordings, measured painstakingly the different ion channels that are expressed along the dendrite over here, and showed that it is not that the cell body is the only place where that nonlinearity that we saw expresses, but that nonlinearity expresses pretty much throughout the entire the neuronal structure. And there are certain ion channels like the Hodgkin, a certain kind of ion channel, KB4.2, another kind of ion channel. These are also nonlinearities that are expressing over here. They were present at higher densities in the distal dendrites compared to the cell basically. So that fundamentally showed that the purpose of the dendrite is not just to collect the information and send it down to the cell, but there is a lot of processing that is going on over here. Right? So I'll show you some functions of Hodgkin which will make you convinced that this acts as some kind of filter bank where the inputs that are coming to the distal locations are filtered at different frequencies compared to the inputs at proximal locations, because the density of Hodgkin is higher here, it's lower here, and therefore there's a lot of processing that goes on over here. And people also found that there were sodium channels here, which meant that you don't just initiate spikes here, you can have dendritic spikes initiated even at this location basically. people have asked questions about what exactly are these ion channels doing over here. And it fundamentally showed us that the law of dynamic polarization that we started out with, that the flow of information is unidirectional, that completely falls apart in real neurons. The action potentials don't go only along the axon, they also come into the dendrite. And the amplitude of action potentials is graded along the dendrites. I mean, usually we thought that it was an all or none phenomenon. You go and measure action potentials here, it will be around 10 millivolts basically. Right? I mean, here it will be a 100 millivolt signal, and down here it might be 10 millivolt, and along the way there is changes. And because there are sodium channels, the nonlinearities that we talked about, they express even in the dendritic locations. You can have spikes here, and you can also have pretty much every single dendrite releases some or other form of neurotransmitters. It's not something which is confined to a specific location. The hippocampal and cortical neurons, they release too many retrograde messages into the extracellular matrix. So every single dendrite pretty much releases neurotransmitters. It's not only the axons, and you don't have them as simple integrating fire. As I said, because of the expression of certain ion channels, they can become band-pass filtering structures, not just low-pass filtering structures, basically. Right? So, how do you account for this? So, you implement cable theory, you take cable theory, implement it. You don't have to solve the second order partial differential equation. What you do is you segment this into smaller compartments so that each of them becomes approximately isopotential, and each of them you can approximate using an RC circuit. And if you know the kind of ion channels that express over there, you can add additional parallel conductances to this, and use that for modeling a neuron in its full glory in terms of electrical computation. So this constituted the third generation of models where you bring in the notion of active dendrites, which expresses these different ion channels, and the fact that each and every input is processed in its own right, rather than all of them being algebraically summated over here and passed through a nonlinearity. There's a lot of processing that goes on over here. How much processing goes on over here? Literally a lot. So this is a paper that came up 4 years ago. So they took a single cortical pyramidal neuron and fed it with several different inputs in terms of spike trains, and recorded the output of that particular thing in terms of voltage, and asked how many layers of deep network do you require for replicating this kind of an input-output mapping. If it were a simple perceptron, like what we saw in the first generation of models, which is what we continue to think it is, then all that you would need is a single perceptron over here, and it should be able to give you the output by just placing a single compartment and action potential generation mechanism over here. It turned out the neuron with the active dendrites with NMDA receptors and other things, it turned out that they required 5 to 8 layers of convolutional DNA for mapping or for approximating to a realistic level the output of a layer 5 cortical pyramidal cell. Right? So that is the level of complexity that we are dealing with, and we are still in the third generation. There is one more generation to come. So the second thing that people realized in the 90s was when the molecular biology revolution came into picture. We think that much of the computation that happens in neurons are all electrical computation. Now, if you read this review and go through this, I've sent the slides already, so you can go through this. I don't want to read this. So you will realize that there are more number of chemical computation, the kind of signaling mechanisms, the enzymes that are present over there, which are responsible for all forms of plasticity and stuff like that. So the number of chemical computations that happens is much higher than the number of electrical computations that happen in a single neuron now. And therefore they started accommodating this into a single neuron model, and because space was already in place, you not just accounted for the fact that there is bidirectional signal transmission and that the nonlinearity is now expressed not just at the cell body, but they are expressed throughout the dendritic structure, but also account for the different signaling mechanisms that are present and how much that they can go along that particular dendritic structure using reaction diffusion formulations, which would also take the second order partial differential equation formulation basically. So that was the third generation where we realized that dendrites are not passive and that the chemical computations outweigh the number of electrical computations per second and they play critical roles not just in adaptation but also in terms of fundamental neuronal functions. So this was in the early 90s. As I said, two things happened. The patch clamp electrophysiology technique became available. People took painstaking effort to map the kind of ion channels that are present over there. And also the molecular biology revolution happened during the 90s. That's when we realized that a single neuron is not just some kind of electrical computation machinery, but it does much more. And those are the conceptual foundations. And instead of dealing with coupled ordinary differential equations across space, we started dealing with coupled partial differential equations across space. These are the kind of models that we use in our lab for modeling single neuron physiology basically. And so that's the third generation of models. The fourth generation of models, which I think we are currently in now, started in the early 2000s when people started recording from many and many neurons and they also, instead of the computational power became much more easily available and therefore instead of modeling single neurons, they started asking questions, what if, so when they recorded from neurons first of all, they found that each and every measurement that they recorded from the population of neurons was variable. Right? I mean, you measure something like the firing rate of a neuron for say 100 picoampere injection, they would see that some neurons of the same subtype will fire 0 hertz action potentials and others would fire at 100 hertz basically. Well, there was huge variability over there. So and more data became available. I mean, initially the number of labs that we were doing was lesser and therefore the data was minimal. But as more and more labs started doing, there was more data. The heterogeneity and the variability became very, very clear that there was variability in terms of what the function is. And when they also asked fundamental questions about what is the relationship between the components that are mediating neuronal function and neuronal function itself, they found that it was variable across different neurons basically. In some neuron, ion channel 1 would have a major effect. In another neuron, it would have no effect whatsoever. In the early 2000s, Professor Eve Marlow, who is in Brandeis University, she came up with this model, a population of network models for a rhythmogenic circuit. This is a circuit that is involved in digestion in lobsters. And these, what she showed was you can get exactly the same rhythms at a circuit level with very different neurons, with very different ion channels, with very different synaptic cascades. Then fundamentally the question becomes, what exactly is the relationship between the components that make a neuron and the function that it is performing? For people who come from physics and engineering background, which include me, we are used to a certain amount of precision in terms of what exactly specific components do and how they contribute to a specific function. Now you have a scenario, every single neuronal subtype that you can take out one single component. In one neuron it will have a maximum effect, in another neuron it will have a minimal effect. So the fundamental question that was asked at the beginning of this was that there are several different molecules that are acting as subsystems, which we saw was ion channels. There are other things like pumps and transporters, and there are specific functions of single neurons in terms of input resistance or firing rate and things like that. As I said, you can have different components taken out, and some neurons would show exactly the same function. In other neurons, you take out the same component, you will see that the effect will be much more deleterious and you would have the neuron not firing at all, let's say. Right? So what exactly mediates this variability? Where does this come from? What exactly is contributing to this heterogeneity across different neurons. And can we even understand that? Because from a replication standpoint, if we don't know what exactly each component is doing, how exactly do we model a function? Or from a pathological standpoint, if we don't know that this particular thing or the absence of this particular channel is what is causing the pathological condition, how do we address in terms of therapies, how are we going to take it back? Right? So that was the fundamental question that was started, that people started asking at the beginning of 2000s. How do these different subsystems come together to yield collective neuronal function? Right? So, and pretty much all of neuroscience doesn't matter whether it is at the cellular level like we do or at the behavioral level that others do. This is phrenology. The fundamental question can be posed as a difference between a one-to-one relationship between specific structures. So as we said, in our case, those would be specific molecules and specific function neuronal firing, for instance. In this case, specific brain regions and specific behavior. Or is it, random combinations of these ion channels or brain locations can come together to yield collective function. Right? So that's pretty much the question that we have been grappling with for a long time, one way or the other across different scales of neuroscience. And even in our case, when we are looking at single neurons, that is fundamentally the question. Is there a one-to-one relationship between the molecules and the function? Or any combination of molecules can give you the outcome? Like pretty much any controversy in biology, the answer always lies in the middle. And it turns out in this case, there is a specific name for that answer. Right? The one-to-one relationship between structural components and function would correspond to a low level of randomness where there is completely determined relationship between the structures and the function. And if you have random combinations of structures giving you collective function, that would also give you low complexity. And it turns out there is a specific class of system called as complex systems, which are neither fully determined nor completely random, where there is intermediate level of randomness, where it is not completely determined, not completely random. And that is what we are dealing with pretty much across every scale of neuroscience. I'll give you the more examples of this. There are some examples down, right? So this is Gerald Edelman. I strongly encourage all of you to read this paper. It's a short paper. Gerald Edelman got his Nobel for the structure of antibody. He moved over to neuroscience after that. He wrote this landmark review in 2001. We'll come to what exactly that word means in a minute. Degeneracy and complexity in biological system. So in that paper, this is the definition of a complex system. It turns out the definition of a complex system is complex. Therefore, it's something that you deal with. So a complex system may be considered as one in which smaller parts are functionally segregated. I'll give you examples. The ion channels have specific functions. Sodium channels allow sodium to go through, potassium channels allow potassium to go through, and so on and so forth. The pump pumps a certain ion outside or inside. So there is a functionally specialized system that is sitting over there. Across a diversity of functions, ion channels, pumps, and so on and so forth. But also as one that shows increasing degrees of integration where more and more of its parts interact. You wouldn't get an action potential if the sodium and potassium channels did not interact with each other. Put otherwise, a complex system may be viewed as one that reveals an interplay between functional specialization, which is this part, and functional integration, which is this part over here. If you codify this as a figure, you can consider these as functionally specialized subsystems. And you could consider these as ion channels, pumps, and other things. And there is, increasing levels of integration, interaction between them. Those interactions are not random. Those interactions are not fully determined. There is specific quote-unquote motif-based interactions, which will come in a minute, between these components, and that gives you collective function. It is as though you are composing the function using the components that are present over there. So let's consider this as a graph. Let's consider these elements to be be the different elements that are present. In this case, it could be molecules. And there are specific kinds of interactions between these components that are present over here. Right? A graph, as all of you probably know, is a set of objects called nodes. Those are the nodes of the vertices. And some pairs of them could be connected by what are called as edges. It could be an undirected graph or it could be a directed graph. So if you represent complex systems as graphs, the connectivity among all the components of this particular graph would neither be all-to-all or a specific kind of connectivity, or it wouldn't be completely random for you to get the outcome. It would be along this intermediate level of randomness. So what do I mean by this intermediate level of randomness? So these are all randomized networks. Let's say that there are 16 nodes over here, and you connect them randomly, generate several different randomized networks, and ask yourself if you find this specific subgraph with this kind of connectivity, A, B, C, A connects to both B and C, and B connects to C, there are no other connections. You want to find that particular connectivity across these randomized networks. And you would see that that kind of specific motif, which is a specific kind of connection, would be prevalent at lesser levels in randomized networks, but in real biological networks, you are in a real complex system. Let's keep biology out for a while. In real complex systems, you would see that that motif would be prevalent at much higher levels, basically. Right? So that is what determines, or that's what defines the intermediate level of randomness. You are neither completely random, where you have randomized connections, or you are not like specifically connected in a certain fashion. You have an intermediate level of randomness in this particular structure. People have found several different motifs in terms of feedback loops, in terms of interactions from one to several, lateral inhibition, cascades. There are several different kinds of motifs that have been found, and this has been found across every single scale of biology. One of the most well-studied ones is that the genetic end of it by Yuri Allen, two very good books on the topic, and the transcriptional networks, and where you consider the two nodes to be genes and the interactions between them as nodes. So that has been studied, well studied at the genetic end of it. In the molecules end of it, you have several different reviews and papers on signaling network, metabolic pathways, and so on and so forth. I'm going to skip cells because there was not much until recently. If you come to networks, again, if you consider neuron to be nodes and the synaptic connections between them to be edges, then you would see that there is feed-forward excitation, feed-forward inhibition, feedback inhibition, mutual inhibition, lateral inhibition. Each of this are the kind of motifs that we have been seeing. It's not like the connectivity between the different neurons that are sitting in the brain is completely random or fully determined. You have an intermediate level of complexity where there is a higher prevalence of these kinds of motifs that are present over there. Right? You could go to systems level and you could look for motifs across network of networks. You would see that there are networks of network, and at the behavioral level it's even more well studied in terms of where scenarios where there are the nodes will be two different animals, and interactions would be like predator-prey kind of interactions, and how exactly it affects the ecosystem and other things have been very tested. Right? So you have these kinds of connectivity where X to Y represents either transcriptional network connectivity, neuronal network connectivity, or ecological food web. But I mean, there was something that was bothering us, that there is nothing at the cellular end of it. Why is there is nothing at the cellular end of it? Is it because there is nothing complex about it? There are no motifs that are present over here? It turned out that, I mean, you had to look across two different scales for finding motifs here. Unlike here, where the edges are all belonging to the same scale. Here, both of them are genes, both of them are neurons, both of them are animals. But here, you would have to go across two different scales, where membrane voltage and calcium and firing frequency would be here, and the different molecules and enzymes would be over here. I'll give you an example. The same example that we saw from the second generation of models, where we said that the rising phase of action potential is because of sodium channels. forget that it was a sodium channel. Think of it as a positive feedback loop, right? A fast positive feedback loop followed by a slow negative feedback loop is what is mediating this action potential. Right? And there is the inactivation that I talked about that is there in terms of auto-regulation. So there is an interaction between two different motifs, and if you look at this, all the reds are going to be cellular, all the blues are going to be molecules. So the specific component that is implementing this is immaterial. All that you need is a positive feedback loop and a negative feedback loop. In the heart, for instance, the rising phase would be predominantly calcium, and there would be several potassium channels that mediate this. But you could think of this as two different motifs interacting with each other. Dennis Noble has been talking about this cross-scale interaction, and he calls it as the Hodgkin cycle, and uses it elaborately for talking about how that's a fundamental thing across all of biology. So that's not something which is limited only to action potential generation. I'll give you some more motifs. So if you want a burst generating neuron, you need a mechanism for sustaining this, and you need a mechanism for shutting it down. So that is the one that sustains the burst, and that's the slow negative feedback loop that shuts it down. Again, the molecules that are implementing this are immaterial. The motif is what is important. That is high frequency adaptation, similar kind of motif. This is graded persistent activity, which is found in working memory systems. Here also you have a calcium dependent feedback loop, but here it is a positive feedback as opposed to here, which was a negative feedback loop, which gives you this kind of behavior. And we talked about that Hodgkin channel, which I said I will come back to this. So this implements a slow negative feedback loop. And so this is a hyperpolarization activated invert current which depolarizes the cell. This is a potassium channel. It's a depolarization activated outward current which hyperpolarizes the cell. Both of them implement a slow negative feedback loop, and both of them, when they express in neurons, give you resonance basically. So this is around 1 Hz, that's around 15 Hz, and you see that this neuron maximally responds to around 6 Hz signals at the chirp stimulus, which we typically use for characterizing these. So if you have these kind of slow negative feedback loops, they suppress the lower frequencies and therefore give you resonance in this particular structure. So that's the presence of a motif. So and when you have again a motif which is implementing a slow negative feedback loop, another that is implementing a fast positive feedback loop, you start getting these subthreshold oscillations. Again, this is something which is prevalent. You can replace this with the Hodgkin, you will still get oscillation. As I said, you can think of these as motifs rather than thinking of them as specific components. And that shows you that there are these motifs that are present in single neurons as well. So if you think of this, you can think of a neuron as a collection of voltage or calcium related motifs. But as we saw, neurons have dendrites, which means that each and every compartment, not every neuron, is made up of several different motifs that are sitting over here. Right? So you could think of a neuron as a conglomeration of different voltage and calcium dependent mechanisms implementing these motifs that are shown over here. These are different ion channels or molecules or pumps that are sitting over here, and they also talk to each other with these kind of mutual dyad motifs across these compartments. And if you have a neuronal morphology that is branching, you will have branches, and that defines how exactly the neuron functions. It's not just across morphology, across the set of components that are present over here also, you are defining what is present over here with versus what is present over here. And that kind of a motif-based interaction is what is giving you the function of a single neuron. Right? So there are non-uniformities in the motif strengths. So the slow negative feedback loop is if you look at the cell body which is here versus the dendrite which is here, the cell body has lower A-type potassium channels, which is the KV channel that I am showing here. Weak feedback, and therefore the action potential amplitude is high. Here the back-propagating action potential amplitude is low. Here the resonance is weaker because this is weak feedback. Here on the other hand, the density of these channels is higher, and therefore the feedback is stronger, and therefore the suppression here is stronger, and therefore the resonance shifts towards the right. So even within a single neuron, you have differential expression of these motifs and their strengths, and even with reference to calcium, that is true. So that is one aspect of what defines a complex system. So where you have this expression of these motif-based interactions, and we saw that across different scales, you have these motifs prevalent. The second and most important thing which explains all that heterogeneity that I was talking about is what we call in biology or in physics also, as degeneracy. It doesn't have anything to do with neurodegeneration or anything like that. It's a fundamental feature of all complex systems. It's the ability of elements that are structurally different to perform the same function or yield the same output. What I started out with was I took one specific set of these functionally specialized systems, introduced one set of interactions based upon certain kinds of motifs, and told that that gives you the collective function. It doesn't need that specific combination. It can pick up another combination of the functionally specialized subsystems and give you the exact same functional outcome. Degeneracy is not redundancy. Redundancy is a scenario where the same function is performed by identical elements. The usual example that I give is the laser pointer. If the battery is dead, I would remove the battery, replace another battery of the same kind, use the same thing. That is redundancy, right? Instead of that, I go to the PowerPoint, pick up the PowerPoint laser pointer option, use that for pointing to specific location, or pick a stick and use that. That is degeneracy. The important distinction is that distinct structural components are required. Right? It's not a one-to-one relationship, and it involves structurally different elements and we yield different functions. Right? So this is a table from that Edelman and Gary paper, and you will see examples all the way from genetic code to interanimal communication in terms of language over here. Right? And once you place it within the motif framework, once you place it within the complex system framework, defining degeneracy becomes much more easier. Right? So here, this is what we call as component degeneracy. This is the same negative feedback loop I said. So you can either have a depolarization activated outward current or a hyperpolarization activated inward current. It will give you exactly the same functional outcome. These are two completely different molecules made up of very different amino acids, very different folds. This is hyperpolarization activated, this is depolarization activated, but still, in terms of the functional motifs that they implement, they give you the same functional outcome. So that is component degeneracy for you. The second one is what distinguishes or defines the neuron-to-neuron variability. Here, the set of components that are expressed are exactly the same for these three neurons, but the strength of that, the presence of one channel, say for instance this particular channel over here is weaker here, is stronger here, is weaker here, and that is what we call as edge degeneracy. The strengths are different over here, and that is why you can explain, say for instance, if you take out that particular channel from this neuron, it might not have any impact over here, but here it would have a strong impact. This has been over and over found across different systems. And a third kind of degeneracy is what we call as motif degeneracy. Let's say you want bursting in neurons. You can get it with calcium activated potassium and calcium channels with this kind of motif. You can just get it with a T-type calcium channel with this kind of motif. You can get it with a combination of two channels like this with this kind of motif. Or you can have interactions between two different compartments in a neuron and you can get bursting over here. And look at the motifs over here. They are very, very different, but the final outcome is exactly the same. But still, there are examples of how exactly bursting is mediated in several different neurons using each of these different examples. If you think that, okay, in this neuron, bursting is implemented like this and go and search for it over here, you will not find it. Right? Or if you say that, okay, it is CAV3, let me block this in this neuron, there is no CAV3 and therefore there will not be anything happening over here. Even though the functional outcome is exactly the same, the motifs are different. So that's what defines this basically. Right? So I'll give you some examples from our own lab in terms of such degeneracy. So we saw that on the same dendritic arbor, you have the backpropagating action potential is higher here, lower here. The here, higher here because of the different motifs that are present over here. So the question that Rahul asked a long time ago, more than a decade ago now, was can you implement all these different functional maps? The backpropagating action potential amplitude should go down, the resonance should go up, the input resistance should go down, and so on and so forth. Can we have all of this across the dendritic hardware with several different kinds of combinations? So that was the fundamental question he asked. And these are examples of neurons where you see that the functional maps are exactly overlapping with each other, at least for most part of it. You see that they are overlapping with each other. But if you look at the parameters that defined them, the parameters that gave rise to those 6 models in terms of ion channel densities and their half maximal voltages, you see that huge heterogeneity that is present over there. Right? I mean, and therefore, let's say for instance, if this is the conductance for the P-type calcium channel, if you take out the P-type calcium channel from this neuron, it will not have any effect. But on the other hand, if you take it out from this neuron, it will have a maximal effect basically, and that is where the variability comes in. Right? So this shows you that you can get exactly the same function with very different ion channel combinations, and therefore you don't have to maintain individual channels at specific levels for you to be able to get the function basically. We have repeated this over and over again in different parts of the hippocampus. So that was the CA1 pyramidal neurons. This is the DG granule cells where there are a bunch of different ion channels, several different measurements on which they were constrained, and imposed them on a morphologically realistic model, and showed both from a computational perspective and using electrophysiological lines of evidence that there is significant amount of degeneracy that is present. Why is this not belonging to here? So when we do this, what we do is we do a stochastic search on this parametric space, and we search like tens of thousands of models. We will see that 99% of them will be false basically. Only 1% of them are valid, and they are not clustered around one particular location. And therefore there is not one single model that is valid which rules out this end of it. If it were random combinations of ion channels giving you this, then all the models should be valid. That also goes out over here because only 1% of the valid models are there. So therefore it falls somewhere in the middle where there are several different combinations that can give you the same functional outcome basically. Right? So this is in bursting neurons in the CA3. This is in the oscillatory neurons that are present in the entorhinal cortex, and where we employed even a specific kind of motif, which is a combination of fast positive and slow negative feedback loops for explaining these kind of oscillations, and showed that there is stochastic resonance as well in these kind of structures basically. Right? So in each and every of these cells that we have looked at, we have found strong evidence for the presence of degeneracy in these structures. So fundamentally, if we go back to the question of how exactly is this a relationship, it is definitely not one-to-one. There are a bunch of functionally specialized subsystems, and you have specific kind of interactions that are essential. You can't have random interactions between these components and expect it to give you the collective function. There is not one single way. There are several different ways by which you can achieve this. And we have also extended this to networks in terms of cells as subsystem and networks as the interactions between these different cells giving you the outcome. Continuous attractor networks, recurrent neural networks, the DG granule cells, specific functions over there, and there also we have specific lines of evidence to show that you have degeneracy there as well, where interactions between these different cells gives you the collective function, not random interactions, not one single combination that gives you the outcome, but several different things. So if you think about it, you have several different molecules and there are cellular scale interactions, several different combinations can give you the outcome, that is cellular scale degeneracy, and synaptic interactions like what we saw over here, giving you network scale degeneracy, and at the systems level you could have different systems interacting with each other giving you the same functional outcome. So there is actually a cascade of degeneracy that is present over here, where at each and every level you could have different combinations of different things giving you the same outcome. Finally, for a few minutes, I'll just talk about the adaptive part of it. My title, the model I said is a complex adaptive system. So one of the things again we assume coming from engineering side again is that the only things that change in a neuron are the synapses basically, right? But in reality, it's actually a multi-component plasticity that makes a brain function. The classical view of learning and memory is that it is synaptic plasticity that drives learning and memory. But in today's world, we know that plasticity is ubiquitous. You can of course change synapses, but that's not confined to that. You can change the excitability properties of the different cells that are sitting over here. You can change the astrocytes, you can change the axonal properties, you can change the density of the extracellular matrix and so on and so forth. But I mean, if everything can change, how do you even maintain stability of the system? It turns out ubiquitous plasticity does not necessarily mean arbitrary plasticity. There are strong constraints on what components can change together. I mean, Poonam came up with this analogy, which I like very much. So you have a cube, and your final goal is this. Let's say this is the learning objective, basically. So in this configuration, you have specific constraints on these components that they have to change together. You can't have this and that changing together, for instance. You will have to move this entire phase together, and therefore in this configuration, these are the components that can change together. In another configuration, there is another set of components that can change together. And for reaching over here, there is not one route for you to be able to come from here to here. You can move different things and eventually reach this particular location. Right? So there is strong structure in terms of which components change together. And there are several routes which we call as plasticity degeneracy in terms of how you arrive at the final solution. I'll give you some examples. So this is what is called the theta-verse pairing protocol, which was thought to be a LTP protocol which changes only synapses. But when people actually in the 90s went after the 90s when people were able to record from dendrites, when they went and recorded from the dendrites, they saw that it was not just the synapses that were changing, the ion channels that were present there also were changing basically. For the same protocol, no changes in the protocol. What mediated this? There were strong network motifs at the signaling level where the calcium that was coming in was activating specific enzymes, and those enzymes were not affecting only the synaptic properties but also the different ion channels that are sitting over there. And this structure is what makes this multi-component plasticity possible. Right? And there is a specific structure. It's not like arbitrary things are changing. There is a specific direction in which they change, and that is imposed by these motifs that are present at the cellular scale. This is true even in the DG granule cells, where three different channels change together basically. And this is theta-verse firing, as I said, three different channels changing. This is even true for the circadian rhythm, where there are neurons in the suprachiasmatic nucleus which go from high firing rate to low firing rate, and it is a cycle, and there are multiple ion channels that change together for you to make this possible. What Harshit did was ask the question of if you have heterogeneous neuronal populations in the suprachiasmatic nucleus, how do you go through this? I mean, how do you, and if you have so many things changing, how do you even go through this? Cutting a long story short, what he found was that you can go from day-like neurons to night-like neurons through several different routes with several different combinations of this. And you can start from the day-like neuron, go to the night-like level, come back, you will not have exactly the same set of ion channels. You will have changed everything, and you would still have the same firing rates, but the kind of channels that are involved changes basically, and there is a specific structure on which these changes occur. It's not arbitrary. So you could even think of plasticity as a complex system where, let's say, I mean, fundamentally there is a change in environment and the system has to adapt to that change. So you want to make sure that your response properties are adapting to that by having a rightward shift in the cumulative thing, assuming efficiency basically. I mean, in parallel you want to maintain homeostasis. But there is not one multi-component plasticity combination that can give you this. You can have plasticity degeneracy where you can have different components changing in different directions depending upon the state of the neuron now, and that can give you the final outcome that you want, basically. Right? So there is plasticity degeneracy and plasticity, manifolds that are present over here also. So the origins for the complex adaptive system was in the early 2000s. This came from the variability, the heterogeneity, the degeneracy perspectives. And it came because large-scale recordings were available. I mean, when techniques were difficult, when you were unable to do this in many different things, you're doing it from one cell and making conclusions from there. And we also made mistakes like averaging across different cells and things like that. The ability to build population of models, big data analysis, threw up all these heterogeneities and different ways of getting through this. I mean, we would have, if we were doing these models in the 1990s, we would have been working with one single model basically. Now, I mean, when people in the lab run 10,000 models and out of which 100 of them are valid, we take that out and analyze them. Therefore, that gives us the perspective in terms of how we can match the heterogeneities in our model population with the heterogeneities in the actual system and what contributes to that. You can go and test that back in the system because we have an electrophysiology lab and ask questions about whether these predictions are workable or are they valid. So we can go back and forth and that is what gave us this complex system framework for neurons. The good thing about this is that it encompasses all the previous generations in one go. You give me any physiological phenomena, I can explain it with the complex system framework, basically. In terms of the heterogeneity, in terms of the degeneracy, in terms of the variable dependencies on the components, all of that can be explained. More importantly, I think for this audience, it also is important to move beyond the first or even second generation of models and move to accounting for space as well as this complexity in terms of the ability to compose functions from the functional subsystems that are present. Instead of, one of the standard cliches about complex systems is that a Boeing airplane is not a complex system, it's a complicated system because there are specific components doing specific things. It's not a complex system. Here you have the functionally specialized subsystem. You'll have to be able to compose a specific function out of these different components, interacting with each other, and it is not a one-to-one relationship. If you might even think of it as an over-complete representation where you have different things that you can pick up and assign coefficients to it and come to the final location that you want to get to, rather than having an orthonormal representation where you have one specific coordinates for that. So that's what this is. So if that is what gives you the robustness basically, because you are not that dependent upon one thing failing. If one thing fails, you don't really care about it. Something else comes up. You stitch the function again based upon other components that are present over there. That is the one that gives the kind of efficiency, the continual learning capabilities to neurons, because you don't have to pick the same resources that were allocated for the other memory. You pick up something else and stitch together the function for encoding this memory. So that robustness comes from this complex system framework, which you wouldn't get from those one-to-one mappings that we have been dealing with for the past century, basically. I mean, this century, thanks to the big data and the big scale analysis, we have this framework, and I hope that people take this forward in terms of building models as motifs, because this is neither molecules nor cell, this is an intermediary, and you can stitch together these motifs for you to be able to get specific functions. Those are the different complexity degeneracy lines of work from the laboratory, much of it based on the hippocampal formation, different brain regions, different characteristics, different physiological properties, different encoding characteristics. There are a bunch of useful reviews from different perspectives. That's from the learning perspective. This is from the efficient information coding, from the Horace-Barlow perspective, not the energy efficiency perspective. We also put together this review on the role of degeneracy in epilepsy. If you can get physiology through multiple routes, you could also get pathophysiology through multiple routes. So that's what we show over here, and you can also repair it with multiple routes. inputs. So that's the network motif that I present.